Hi, this is Devpratim from IITGuwahati. I am working in the Department of Chemistry. So, recently we developed a new peptide based hydrogel for efficient delivery of drug. So, I'll talk about it today. But before that, I'll just mention what's the problem, the current situation is. It's like cancer is it's a well-known disease, and it's one of the biggest problem in the world right now. But what cancer is actually, it is uncontrolled and unwanted growth of cells. So the treatment for cancer is to kill these unwanted cells, and for that we have plenty of drugs in the market. So we know which molecule or which drug can kill these cells, but the problem is when we administer that drug in our body, it also kills the healthy and growing cells. So you might have seen in your family and others, whenever the patient gets chemotherapy, the problem what they suffer is the hair loss, the nails get fragile, and there are several other side effects, like all the other organs starts getting damaged because of this toxic nature of the drugs. So for that, the solution is that you deliver that drug to the tumor, directly to the tumor. Plenty of ways people or scientists across the world are trying to do that. I am not going into the details of all those things, but one way is to use these hydrogel based systems, which are very soft gels. But the problem with hydrogels is that you cannot administer semi-solid hydrogel in your bloodstream. So for that you need a gel that can be injected, means when you press the gel it becomes liquid, and once the pressure is gone it becomes gel again. So if you can make something like that, some sort of gel like that, then only you can load the drug in that gel and deliver on the organ. But there are several other issues, that once the gel goes there and you put it on the organ, then it slowly gets dissolved in the biofluid. So not all gels can be used. If you can make something, a gel which can be injected and doesn't get dissolved, then the problem is how the drug will be released. So in 2018, 2019, at that time, we actually developed a very small peptide based gel. We call it PyKC. Interestingly, we found that the gel is insoluble in biofluid like human blood serum or any aqueous medium. Also, another important fact is that the gel slowly gets broken in presence of glutathione. Now, glutathione is a molecule that is overexpressed in cancerous cells. Means near the cancer cells or tumors, there will be a significant amount, high concentration of GSH or glutathione. So once we developed that gel, we realized that it has the potential to be used for localized delivery. But the problem was whether this gel is injectable or not. Then we analyzed the mechanical properties of that gel, and we found that yes, it is very much injectable. And then we discussed with people from Bose Institute in Calcutta, because we don't do this animal studies here in our lab. So in collaboration with Dr. Kuldeep Jana and Satyajit Haldar from Bose Institute, we started our journey on mice model. And to cut the story short, what we found that it is working excellently, and the normal dose what a patient gets, or the mice, based on the body weight they get, we used exactly the same amount of dose, loaded it in the hydrogel matrix, and then injected it on the breast tumor, and we monitored the mice health and everything. After 18 days, what we found that the tumor is reduced by 75%, which is a significant success in this field. You can just imagine that with a single dose, the tumor got reduced to 75%. it's a very time consuming experiment and it takes a lots of time. You can understand it took almost 5 years for us to complete this story, though it's not complete yet, because we don't know with a single dose to what extent maximum the reduction will happen, because after 18 days we studied that and we found 75% reduction. If we study after 30 days, it may be completely gone. That study we couldn't perform because of the lack of time. That that study is going on, but overall what we found that with this excellent small gel, which is absolutely biocompatible, there was no side effect that was observed on any of the organs or the overall mice health. This can reduce the tumor by 75%, as I said, and after 18 days when we cut it open, we found this reduction in the size, and also there was still some gel left with the drug. That means it will release the drug slowly, even maybe another couple of weeks, and by that time the tumor may be absolutely gone. So this is the story what we developed, and we recently published it in Materials Horizons, one very high impact journal from RSC, Royal Society of Chemistry. Thank you.
My name is Ritvika Kushwaha. I am a 4th year PhD student from the Department of Chemistry, IITGuwahati.
I am Tanushree Das, and I am doing my PhD in IITGuwahati, and I am a 5th year student. So in this work, we have developed a low molecular weight based dipeptide gelator. So it forms a hydrogel in basic medium. So although it's a supramolecular hydrogel, but it is insoluble in water, but it can be solubilized in presence of GSH. So you all know that in cancerous cells, so cancerous cells are overexpressed with GSH. So our initial idea was to whether we can use this hydrogelator for cancer treatment. So here see.
So like there is problem with the regular gels, it cannot be easily injected inside the system. So through studying the mechanical properties of our PiKC gel through using rheology, we realized that this gel can be injected. So we loaded it with doxorubicin, and this drug loaded gel, we anticipated that this gel can be used for localized drug delivery to treat cancer cells.
So in our lab, we did the in vitro study where we loaded our hydrogel with doxorubicin, and we checked the release of this doxorubicin from hydrogel in presence of GSH, and we observed that this hydrogel can slowly and sustainably release this doxorubicin, and next then we confirmed that this can be used for this cancer treatment. So next, this work, whole biological work was done in Bose Institute, and this is done by Satyajit Haldar and Professor Kuldeep Jana. Thank you.